Welcome, welcome to the review.
Maybe we should wait like a
Couple more
few.
minutes? Yeah,
Yeah.
it's a good
I'd say
idea.
like 5 minutes, 5 minutes.
Mhm.
I don't know if they realize it's 6. Yeah. They probably thought it was gonna be 7 or something.
Yeah. Okay, 4 people. Let's go. All right, so we're gonna go ahead and get started. This session, this review is gonna be a little bit different than last time. We're not gonna do breakout rooms anymore. So if you want to review the video, the video will have the questions and the Q&A session. So we're just gonna do the Q&A session all in the same room, Zoom. Okay, so let's begin with topic 6. Let's talk about the difference between osmosis diffusion. So both of these are like basically a form of passive transport. Osmosis specifically refers to the passive transport of water towards equilibrium. On the other hand, diffusion, you're gonna just it's referring to particles like glucose, sodium, potassium, things like that. And as usual, or I'm sure this is a review for you guys from Bio 2, things will move from high concentration to low concentration. And in terms of water, it's going to move from low osmolarity to high osmolarity, which we will go over. So diffusion, right? There's always going to be a driving force for it. You're going to often hear electrochemical gradient, which refers to both a chemical gradient and electrical gradient. These are basically supply the energy to allow these particles to move, despite the fact that they don't use energy. They don't use ATP, but in terms of how they move, this is what is actually driving the movement of these particles. Of course, there's also other factors that affect diffusion, and it's J is net flux. Think of net flux as simply the rate of movement. Permeability, distance across the membrane, and concentration difference, they all affect it. I'll talk more about that in a bit. So one of the main things you need to understand for this exam is predict the net diffusive force on a particle and also predict the state at equilibrium. So like we said, in a chemical gradient, you're always going to move from a high concentration to a low concentration, right? So in this case right here, we have sodium. There's only sodium on the left side, none on the right side. So there's going to be a chemical force to the right. All right, but now let's say we add in this anion here. It's still not going to affect the chemical gradient, right? There's still only a sodium on the left side, so the chemical gradient will still produce a force towards the right. However, since we added this anion, what we actually did was, let's say we alter the charge now. On the left side, we have 35 millivolts, right side, negative 35 millivolts. So the electrical gradient, as you know, you probably know this, opposites attract, right? So in this very specific example, the chemical gradient force is to the right and the electrical gradient force is also to the right, okay? So very simple so far. Where it gets maybe just a little bit more confusing is when we have to predict the equilibrium state. So let's just say in the first example where only the sodium was in this beaker, let's ignore this this ion here. What would occur is there would be equal concentrations of sodium on both sides, right? Obviously that's equilibrium, okay? Very simple. On the other hand, let's predict the equilibrium states if we also take into account the electrical gradients. So what actually ends up happening is, let's think about it this way. How would we be able to make the whole beaker a neutral charge? We would have to transfer all of the sodium on the left side to the right side, right? And then the sodium charge would cancel out this anion charge, and then the whole beaker would be neutral. Obviously, that can't occur because what first happens is, as we can see, the chemical gradient and the electrical gradient are pushing the sodium to the right side, right? And like I said, the only way to neutralize this negative charge is if we transferred all of the sodium over. However, eventually, after, let's just say we reach half sodium on the left side, half sodium on the right side, the right side is still a negative charge due to the present here, right? So after chemically you reach equilibrium, the negative charge still exists. Because it still exists, there's still an electrical force to the right, okay? And therefore, after the chemical gradient reaches equilibrium, the electrical force is still acting. So you're going to have more sodium on the right side of the beaker. And then once again, the right side is still negative. It's just slightly less negative, but the left side is slightly less positive, okay? And there will be a practice problem, I think, on this as well. So now let's talk more about diffusion or passive transport. There are 3 different types you should be aware of. Non-polar, hydrophobic, small particles like oxygen, like carbon dioxide, and even water. Water can actually diffuse through here. But over here we have channels, ion channels, and also aquaporins, which expedite the movement of water. And then here we have specifically facilitated diffusion or transporters. And so the main thing we need to understand here is, I already said those. Okay, so first let's talk about this equation, sorry. Notice that symbol indicates flux is proportional to all of these things over here. So basically an increase in permeability will increase the flux, increase in your surface area will increase the flux, and a greater concentration difference will increase your flux, aka your flow rate. However, something to note is that while simple diffusion and while channel diffusion, no matter how great the concentration gradient is, the net flux will be able to increase. Okay, this means it's not saturable. Same thing with channels. No matter how great your concentration gradient is, your net flux will increase. On the other hand, in facilitated diffusion or transporter diffusion, what you have is these proteins act as enzymes. They have active sites. And think of it like a bus. A bus only has so many seats for so many molecules to transport. Eventually, all the active sites will be filled, and no matter how large you make the gradient, no matter how many people or molecules you have on this side, the speed will not be able to increase. And you call that saturable. So this is important because on an exam, maybe in the figures he gives you, there's going to be a bunch of different lines, and you should be able to pick out which one represents simple diffusion, which one represents channel diffusion, which one represents transporter diffusion. And also, just remember that this does not use energy. This is once again following electrical and chemical gradients. Let's move on to active transport now. We have primary and secondary. So obviously in active transport, you can do two things. You can transport molecules against their gradient. For example, you can pile up sodium on one side of a cell while creating, a deficit on the other side. And you can also increase the concentration past equilibrium, which passive diffusion cannot do. Okay, so that's when you see a graph, maybe a figure where it has a control.
I think your mic cut off.
I'm sorry about that. Yeah, so think of active transport when you see this upward shift in this curve here. And obviously equilibrium is where the concentration, or sorry, not the concentration, where the net electrochemical force directing inwards is equal to the net electrochemical force directing outwards. So concentration does not always have to be equal. If charges are involved, you have to look at the net force, not just the concentration, all right? And here in active transport, we're simply able to we increase a concentration gradient more than it would normally be at in equilibrium. Or yeah, normally more so than where the net forces are equal to each other. These are examples that you should know. He may reference them and he may ask you specifically what they are transporting. The NHE, I believe, sits in your stomach. I don't recall exactly, but make sure you review that. You need to know all of these. Know the SGLT, know that it's a co-transporter for sodium and glucose, and obviously NKCC, NCC as well. And obviously these first two as well. So make sure you review your notes for these. You should understand their function and their location. And so now let's talk more specifically how everything interacts with everything. So the sodium potassium pumps is responsible for generating this large sodium gradient, right? There's a excessive amount of sodium on the outside of the cell compared to the inside where there's actually more potassium. Okay, so, and the reason why that's important is because, right, that actually, this generation of a gradient allows for the coupling of sodium passive diffusion with unfavorable transports. So once again, this active pump, which directly uses ATP, it directly uses energy to create a high concentration gradient on the outside of the cell. Secondary active transport couples this concentration gradient with another unfavorable transport. Notice the arrows and how they specifically, a dashed arrow represents diffusion, while a solid arrow means you're going against the gradients. And then also up here, we can see our examples of facilitative fusion. Once again, these do not use energy. They are saturable, and they travel down their concentration gradients. And then this is the example Dr. Cho likes to use. The sun represents the sodium potassium pump, which basically creates this huge reservoir of water. The water flowing through the dam represents the passive diffusion of sodium. And then we're coupling the diffusion of water with the generation of electricity. And that represents glucose being transported into the cell. Okay. And so now let's talk about osmosis. In comparison to when we're referring to how, particles like sodium move across the membranes, you only look at sodium concentration. For example, when we were determining the chemical gradient of sodium, we did not take into account A minus. A minus contributes to the charge, right? But it doesn't matter how much A minus there is, as long as there's sodium on the left and none on the right, we're gonna have a force to the right, okay? However, when you talk about water, you actually have to take into every single solute that is dissolved in that water. And so in this picture here, what you can see is, potatoes placed in salty water, and the water flows into an area of high osmolarity. And so, and more specifically, just as a reminder, I specifically said that you have to take into everything that dissolves in the water. So NaCl will actually break down into sodium plus ions and chloride ions. So you actually have to multiply the osmolarity of NaCl, or the concentration of NaCl. You're gonna have to multiply it by 2. Look, if we look here at magnesium chloride, there are 3 different particles that will dissolve in the water, so we'll multiply that by 3. And obviously, 1 molar equals 1 osmolar, and so it's just 1 to 1. Okay, and as you can see in this picture here, water flows from low osmolarity to high osmolarity. So it's the opposite, not intuitive thinking compared to the chemical gradients. Okay.
All right. Should I share my screen for this one?
Yeah, sure.
All right. Can you see my screen?
Yeah, it looks good.
All right, cool. Okay, so I will be the topic expert for topic 7, and I'm going to be talking about cell, blood salt, and water physiology. Okay, so for this first topic, I'm going to elaborate a little bit more on the osmolarity. So what osmolarity is it basically quantifies the tendency for a solution or a cell to gain or lose water through osmosis when placing it next to another solution. So what I mean by that, so let's say if something is hyperosmotic, it actually has a higher tendency to gain water since it is so concentrated with all these solutes. Whereas if it is hypoosmotic, it has a lower tendency to gain water, or you could reword that and say it has a higher tendency to lose water through osmosis since it is not as concentrated with solutes, right? Ka already talked about how to essentially determine the osmolarity, so I'm not going to go over that again, but I will go over tonicity. So what tonicity is, something that you should remember, is that it only describes solutions, right? And essentially how it affects cell volume. So in this case, in the middle right here, for an isotonic solution, the cell does not change cell volume because essentially we're assuming that there's probably equal solute concentrations in the solution as well as in the cell. So it's neither going to shrink or swell in terms of the cell. But now talking about hypertonic solutions, right? This solution will be classified as hypertonic because it causes the cell to shrink through the water moving out of the cell. Because there's such a high concentration of solutes here, it drives water movement out of the cell and therefore cell shrinkage. That's why this solution will be classified as hypertonic. For the case of hypotonic, there is a net movement of water into the cell, right? So the solution will be classified as hypotonic because it causes the cell to swell. One thing that you should remember is that most hypertonic solutions are going to be hyperosmotic, and also most hypotonic solutions are going to be hypoosmotic, but that is not always the case, but I will be going over that in one of our practice questions. Okay. Now I'm going to be talking about the problems that may be associated with exposing a cell to hypotonic versus hypertonic conditions. So first off, if a cell is placed in a hypotonic solution, it's going to facilitate water movement into the cell. And if water is moving into the cell so quickly that it causes it to swell up, the cell can actually burst. And if it bursts, obviously the cell is going to die. Also, your brain cannot accommodate such high degrees of fluid accumulation and swelling in these brain cells because this can ultimately result in like collapsed vessels, mini strokes, and essentially this can be lethal. And in terms of inhibition of enzyme activity, you should remember that proteins actually evolved in a way to fold in the presence of specific osmolarities or salt concentrations. So if you have higher water concentrations in the cell, and as a result of placing it in a hypotonic solution, it will inhibit this proper folding of that protein. And if you inhibit the proper folding of the protein, you inhibit essentially the function of the protein. So essentially, if there's no structure of the protein, there's no function in terms of that protein. So now I'm going to be moving on to hypertonic, the problems that may be associated with hypertonic. So similar to the hypotonic solution, it's going to result in the inhibition of enzyme activity, right? So why is this the case? Well, in hypertonic solutions, you're exposing these cells to really high concentrations of inorganic ions. And what these inorganic ions do is that they'll actually end up disrupting all these non-covalent interactions, like these hydrogen bonds, these hydrophobic interactions that allow this protein to fold into a proper conformation. And if the protein can't fold into a proper conformation due to these high salt conditions, you're essentially inhibiting the protein from actually functioning. Also, in terms of hypertonic solutions, it results in protein aggregation. So to think about it this way, if you expose a cell to a hypertonic solution, it causes the cell to shrink, right? And if the cell shrinks, all these organelles and these proteins within that cell are going to be crammed together. And when you cram all these proteins together, you get these unnatural interactions that cause these proteins to stick together and aggregate. Also, another thing that you should know is that exposing it to hypertonic solution can cause DNA strand breaks. The last thing I'll talk about in this slide is the factors driving protein aggregations. You guys should remember that polyglutamine containing proteins are more prone to aggregate and has been associated with Huntington's disease and 9 neurodegenerative disorders. Another factor driving protein aggregation would be aging. So essentially, the older the organism, the older the cell, proteins will aggregate in an accelerated manner. And as I mentioned earlier, hypertonicity, hyperosmolarity, and cell shrinkage facilitates protein aggregation. All right. Moving on, we're going to be talking about the Wink and PASC-OSR1 pathway. It's important to note that this is an acute, rapid compensatory response to changes in cell volume, right? So Wink and PASC-OSR1, these are just fancy ways of saying kinases, right? And remember that the role of a kinase is to phosphorylate molecules, and phosphorylation of these molecules will either inhibit or activate their activity, right? And for the case of this cell, the cell is shrinking, right? Why is that the case? Well, the cell might be exposed to some hypertonic solution that's causing water to move out of the cell, right? So what would be the compensatory response to cell shrinkage? Well, you want the water and solutes to flow into the cell, right? So if solutes flow in, water will follow it and cause the cell to swell up back to its normal shape. So that's really the general basis of this mechanism, but I will go over a step-by-step of how it actually works. So in terms of the first step, this WNK, this kinase, activates PASC-OSR1. And by activation of this other kinase right here, this kinase will actually activate these co-transporters, or not activate, but phosphorylate this NKCC and KCC co-transporters, right? It will activate, or it will phosphorylate them at the same time. When NKCC is phosphorylated, it will actually activate it, allowing sodium, potassium, and chloride ions to flow into the cell, right? Whereas when PASC-OSR1 phosphorylates KCC, this co-transporter right here, it actually inhibits its activity. If you inhibit KCC, preventing solutes K, or potassium, and Cl from flowing out. So essentially what happens after allowing these or after phosphorylating these co-transporters is that you're going to allow a net movement of solutes into the cell, right? And if you allow a net movement of solutes into the cell, water will follow and allow it to swell up back to its normal shape. Moving on, I'll be talking about the other case in which a cell swells, right? So if a cell is swelling, it's going to be full of all this fluid, all this water. So essentially the compensatory mechanism would be to allow water to flow out, right? But how essentially do you do that? Well, you want solutes to flow out as well, because if solutes flow out, water will follow. So essentially how this mechanism works is it's a little different in comparison to shrinkage. In this case, these kinases, WNK and PASCO-SR1, will exhibit very low activity. So if they exhibit very low activity, there's a lower incidence that they'll actually phosphorylate NKCC and KCC, right? So if there's a decreased incidence that will phosphorylate NKCC, there's a decreased chance that NKCC or these ions will flow into the cell, right? There's a decreased possibility that they will flow into the cell because it's not activated, right? Where in the case of KCC, phosphorylation actually inhibits KCC. So if you're not phosphorylating it as much, it's like basically taking off the handcuffs or shackles from this KCC transporter. So now you're essentially allowing this potassium and chloride ions to flow out, right? And for the case of this, the phosphatase, I forgot to mention the other slide, phosphatase actually functions by removing phosphate groups, right? So if you remove phosphate groups from NKCC, you decrease the chance that this will be activated, decreasing the chance that ions will flow in. Whereas PP1 will actually remove phosphate groups from the KCC, or this co-transporter. And by doing that, you increase the possibility that potassium and chloride flow out. So essentially the big idea behind this whole mechanism to cell swelling is that there will be a net movement of solutes out of the cell. And if there's a net movement of solutes out of the cell, water will follow, essentially allowing it to shrink back to its normal size. All right, so now we're going to be talking about organic osmolytes, which is more of a long-term response to high osmolarity in comparison to the Wink PASCO-SR1 pathway, which is more of like a short-term immediate response. So for this case, remember when cells are like shrinking or when cells are exposed to like hyperosmotic environments, water will typically want to move out of the cell, right? To mitigate these like high solute concentrations. But in order to counteract this shrinkage, we actually want these transporters to be activated so that solutes could flow into the cell. Because if solutes flow into the cell, water will follow and the cell will swell back to its normal shape, right? But the problem behind this of essentially allowing solutes to constantly flow into the cell is that they'll be full of these really high concentrations of inorganic ions. And if there's these high concentrations, it can destabilize all these protein structures and cause a series of other problems. So our solution was organic osmolytes, right? So this is something that animals have developed. So essentially how organic osmolytes function is that over time, they'll actually replace these inorganic ions, right? So essentially they'll function in the same way, allowing the cell to have a higher osmolarity, right? So that it doesn't shrink when it's exposed to really hypertonic environments, right? So that water doesn't move out. That's why you want these osmolarities to be relatively equal to the environment so the cell doesn't shrink. But the one thing that's different behind these organic osmolytes and inorganic ions is that organic osmolytes do less damage or they do have little change to protein structure or enzyme function, right? So that's why they're essentially better in terms of organic ions filling the cell. Examples of organic osmolytes that you might see on the exam are glycerol, sorbitol, and urea. And essentially, he'll probably ask you a question that says, like, what's a long-term response or a chronic response for osmotic homeostasis? So if you see something related to long-term, first thing I would look for is something related to organic osmolytes. For the last slide, I will be talking about the phylogeny and essentially how aquatic animals and terrestrial animals exchange with the environment. these are the last slides in that topic 7 lecture that you could feel free to go over again, because this will pop up in the exam for like probably like 1 or 2 questions. So it's good to remember these. So in terms of aquatic animals, how they exchange with the environment, just remember this formula right here. Just know that excretion and egestion of like waste materials and other byproducts right here is equivalent to ingestion plus metabolism and absorption. So what metabolism is it could be like cellular respiration, and the byproduct would be water, and essentially that's incorporated into that organism. And absorption for aquatic animals, this is special to aquatic animals, is that they're going to be in direct contact with their environment and essentially absorbing contents through their gills. So aquatic animals have this capability to absorb because they have gills, right? So just remember this. And in terms of terrestrial animals, they're able to exchange with the environment in similar fashion, but they do not have the ability to absorb because obviously we do not have gills. And so therefore they cannot, interact with the fluids and exchange contents like that. Well, one thing we do have in terms of terrestrial animals is evaporation. So evaporation could be in the form of like sweating or perspiring. So essentially just remember this equation right here. Just know that excretion, egestion, and evaporation is equal to ingestion plus metabolism. In terms of phylogeny of osmoregulation, really the big things you have to remember is that land animals, these terrestrial animals, have a milliosmolarity of 300, as well as these bony fish. Just know that sharks and rays will typically have a milliosmolarity between 600 to 1,000. And remember that these marine animals in the bottom 3 or 4 right here is going to be around 1,000 milliosmoles, right? This is important to note because remember, these marine animals are conforming to their environment. And essentially their environment is seawater. And the milliosmolarity of seawater is going to be 1,000. So that's why they try to maintain or conform their milliosmolarity similar to seawater. The one outlier would probably be lampreys, 300 milliosmoles, but he probably won't ask you a question about that. I guess that should be the end of my slides. So super.
Okay, I'll do topic 8. Let me just share my screen. Okay. Okay, so we'll move on to topic 8 now. So we'll start off with urine concentration versus ECF concentration, basically like the effects. So remember, osmolarity is basically like a measurement of solute concentration in whatever liquid you're looking at. So if something has high osmolarity, it's going to be heavily concentrated. So there's a key idea here, which is what you should be focusing on when you're like taking the exam. And basically like the kidney is able to change the concentration of our cellular fluid depending on whether it can make a dilute or concentrated urine. And this is because like that ECF is what's going to filter through into the tubules in your kidneys, and since you're going to be excreting that out, that just directly affects your extracellular fluid concentration. So basically just remember that the more concentrated your urine can be, which would mean like it has more solutes, it didn't like reabsorb as many salt, the more concentrated it is, the more it can lower your ECF osmolarity, and the more dilute your urine can be, and this would be like there's more water in the urine or less salts, the more it can increase your ECF osmolarity, because that would mean less of the salts are in your urine and they're in your extracellular or interstitial fluid more. So, okay. And then urine volume and blood pressure. So your kidneys, by extracting fluid that comes from your extracellular fluid, they can like regulate your blood pressure as well. And this has to do with like the blood volume. And the blood volume is like directly correlated to like how much water you have in your blood plasma. So if you have a lot of water, that's more pressure on the sides of the vessels, the blood vessels. So having more plasma means you have a higher blood pressure. And so if you have a decreased amount of water in the plasma, then you have like a decreased blood pressure. And once again, this is because like urine is mostly water. And the kidneys, they're able to control the amount of water that is filtered into your urine. They're able to do this through like maybe the amount of aquaporins that are lining like the collecting duct, so they can control if there's like a lot of water that's going to be left in your urine or a lot that's going to be reabsorbed. And then this water is going to be coming from your like blood vessels. And we'll talk about like, we'll go more into detail with the structure, I think after this slide. So basically, the more the kidneys increase the volume of urine by extracting more water into the urine, the lower the pressure inside your blood vessels become. And then the lower the volume of urine, decreasing the amount of water, the higher the pressure inside your blood vessels will become. So it's kind of like an inverse relationship for both concentration of ECF and your blood pressure. Okay, so now we'll be talking about the ultrafiltration in the glomerulus. So basically, as we were saying before, the reason like it can affect this like the way this is all like set up, the way like urine is kind of created is kind of like, we'll be going over that right now, and it kind of explains like how your kidneys can affect your blood pressure and your ECF concentration. So basically, you have a bunch of major vessels, and mostly your arterioles, they lead to the kidney, and they can break down into like little vessels or capillaries, known as the glomeruli. This is, you can think of this as the glomerulus, this entire like capillary system, and it's like there are little slits between the capillaries and the Bowman's capsule. So pressure from your heart, because your heart is pumping blood out to the rest of your body, and this pressure is what pushes the fluid through the little slits in like the blood vessels into the Bowman's capsule. And that's kind of how like your filtrate is created. So most of the fluid that's in the vessels is like from your blood plasma, and most of it is like the same as what you see in your blood plasma, because it's just filtering through. It's just basically moving from one area to another, except like it doesn't include your blood vessels or proteins, because those are too big to go through. It's gonna include things like water, salt, glucose, and maybe sometimes amino acids. So it's going to filter a lot of that through into the renal tubules, and the vast majority of this filtrate, which is composed of salt and water and has to be reabsorbed from the tubule back into the blood. So surrounding like each of these like tubules are a bunch of like other capillaries, and they're very close together, so like you're able to reabsorb a lot of the things from the filtrate in the renal tubule. But then also sometimes there's a very small amount of things that are secreted into the renal tubule from the capillaries, but this is a really small amount, so it's not that like it doesn't really contribute much to the urine, but it's just a really small amount of things. The main goal of urine is to like get rid of your toxins and waste, so those aren't going to be reabsorbed, and your kidneys want to like concentrate those in your urine, which is why they end up being excreted, because you don't want to have like toxin waste in your blood. But obviously, like as I said before, the kidneys can like control how much salt or water they want to keep in the urine. But overall, it's like a majority of it is reabsorbed. Okay, so now we'll be talking about the glomerular filtration rate. So the GFR, it estimates the total amount of blood plasma that's filtered in the glomeruli. And you should think of it as like a starting filtration rate. It's like focusing on like the fluid that filters from the capillaries into the Bowman's capsule. So it's like the beginning of the journey for urine. So it's used to check like how well your kidneys are functioning. And you can like basically, if you have like a high GFR value, which when you do like the math related problems on this, you usually get like a pretty like large value. That's like a sign of a normal functioning kidney. And so there are like a few requirements when you're measuring GFR. You're usually focusing on a specific solute, and in his lectures he kind of mostly talks about creatine, which he'll probably ask the questions on the exam like focusing on that solute as well. But basically, like the reason why creatine is like an example solute is because like it can be freely filtered, and it's not like reabsorbed or secreted once it enters the capsule. So basically, it's small enough to be filtered through from the capillaries so it can enter, and then it won't leave the tube once it's in the tube. You don't want it to leave the tube because when you're measuring GFR, you're going to be collecting urine, and you need the solute to be in the urine to like calculate, to measure the values that you need. And then, okay, so then to calculate GFR, I did get this picture from my slides, but I was told that you guys didn't really do like a practice problem with these values. So basically, we know that like the filtrate that's in the fluid that's in the capillaries that's going to go into the Bowman's capsule, it's going to have the same concentration of the solute. Because there's like no change, there's nothing that you're doing there to get like less salt in, because all of it is just going to go into the tube and then get reabsorbed later. But right now at the starting point, you're just going to have like the same concentration of solute in the fluid. So you can just like maybe like draw blood from someone and be able to like measure the concentration of whatever solute you're looking at. And so that's going to be your initial concentration of solute. So it's the 0.01 milligrams per milliliter. And then once like the person you're looking at, once they've like, you're going to collect their urine, and then you can use that urine to measure the final concentration of the solute. So in this case, it would be 0.67 milligrams per milliliter. And then you can also calculate the amount of urine the person produces like in a specific amount of time and get like the urine flow rate, which you can think of it as like the final flow rate. And in this case, it would be 2.3 milliliters per minute. So basically you have 3 values and you're just missing one value, which is the starting flow rate. So you have the initial concentration, the final concentration, and the final flow rate. So there's like some physics involved in this because this deals with like flow, like the flow of liquid. So in that way, you can kind of think of it as like this equation that I have right here. And in like where the start equals the final. And so you can plug in your values here and then find the starting flow rate. And then another way, if you can't remember, this is the GFR should have units of like milliliters per minute. So you could just like set up the equation based on their units and like cancel things out until you can get milliliters per minute. But that's like a little complex. And I think it might be easier just to look at it as like the starting values equaling the final values, and then you're solving for the one variable that you're missing. Okay, so right after the Bowman's capsule, the urine is going to go into the proximal tubule. So the GFR is much greater here than the UFR, which kind of makes sense. Like you're going to be filtering in a lot more fluid than you're going to be excreting. And because you're like taking in so much like fluid into the renal tubules, most of it needs to be reabsorbed. So that's why like the proximal tubule is where like bulk reabsorption occurs. So a lot of water is reabsorbed, and then a lot of essential solutes, like the ones listed here, they're also like able to be filtered through with the water. So around 60 to 100% are reabsorbed. And I think in his like lecture slides, there was a table that kind of tells you like the percentages of each solute or like water that's absorbed. And it'd be kind of good to know that, because I feel like I kind of remember a question kind of needing that. It's just good to know like how much of each is absorbed, reabsorbed. So now we're kind of gonna go into more detail about like the process of reabsorption, and it deals with a lot of like transporters that I think Ka talked about earlier. So we'll start off with like, I'll just like orient us right now. So we have like the renal tubule. Inside the tubule is the lumen, and that's where like the tubular fluid or like the urine is. So then the fluid is surrounded by the wall of the tube, which like is made up of cells, which is like represented here. And then outside of it is like the interstitial fluid, which kind of like interacts with the blood plasma because the capillaries are really close. So you can think of it like that. So basically we'll start off with like the sodium potassium pump, or like ATPase. So it's going to be using primary active transport to pump out 3 sodium ions and pump in 2 potassium ions. So the major point here is that it's getting rid of the sodium ions. Like it's pushing the sodium ions out of the cell into the interstitial fluid. And the reason why this is important is because you're trying to make a sodium gradient. The sodium potassium pump basically creates the sodium gradient, which is what's going to help. Basically, like that gradient is what other transporters use to do their transporting. So the sodium potassium pump, it creates a sodium gradient. Now the concentration of sodium is really low inside the cell. So the NH can use this gradient to pump out protons. And the protons will come from like carbonic acid. So basically the NH3 transporter is pumping out protons, but it's also like taking in sodium, which makes sense because like the concentration of sodium is pretty low now. So it's pumping in sodium ions, and then it's like pumping out protons out of the cell. As it's doing this, it's also generating bicarbonate, which will end up leaving the cell, going into the interstitial fluid through the NBC transporter. And then the SGLT2 transporter, which is also on the apical membrane, is also using the sodium gradient to pump in glucose into the cell. So both SGLT2 and NH3 are bringing sodium into the cell because the concentration is low, and the concentration of sodium is kept low in the cell because of the sodium potassium pump. And then this glucose can then like leave the cell again. on the basolateral side and go into the interstitial fluid. And it uses like a facilitated diffusion transporter here. And then back to the sodium potassium ATPase. While it's creating a sodium gradient like that, it's also like making this side, the interstitial side of the cell, very positive. So this kind of causes like chloride ions to be able to move from the tubular fluid, like from the lumen, all the way into the interstitial fluid, because like while there are tight junctions in the proximal tubule, these tight junctions are kind of leaky, so they're able to kind of leak through, but this is only in the proximal tubule. So, and then lastly, like overall, you're seeing like most things are moving from the tubular fluid side to the interstitial fluid. So most things are moving left to right, most the solutes. And the thing about water is that it's always going to follow where the solutes are. So it also moves from the tubular fluid side to the interstitial fluid. And that's kind of how reabsorption occurs in the proximal tubule. Okay, so then the distal tubule, also it changes the salt concentration and pH of like the tubular fluid or like the urine. And so it doesn't really deal with like water reabsorption. It has like very low permeability for water. So very little water can get reabsorbed in the distal tubule. It's mostly done to like dilute the urine. Like the distal tubule is what like allows for dilute urine to be created, and it mostly like focuses on reabsorbing NaCl. Okay, so then a little more detail into the reabsorption process. So it's kind of similar, but a little bit more simpler than the proximal tubule. It's more focused on just the solutes. And it's kind of like the same orientation in this picture. We have the lumen, which is like where the tubular fluid is, the cell, and then that lines like the tubes, and then the blood or interstitial fluid. They're not the same thing, but you can just like kind of think of them as like extracellular fluid for now. So just like the proximal tubule, there's also sodium potassium pump on the basolateral side of the cell. And it does like the same thing. It has the same purpose of creating a sodium gradient and keeping the sodium concentration in the cell low. And this kind of helps, this is like done so that the NCC transporter can do its job. So the NCC transporter is on the apical side of the membrane, the apical membrane side. And so the sodium gradient, it uses that sodium gradient to move the sodium and chloride ions into the cell. And then we can pump sodium back out through the sodium potassium pump. And then we can like move chloride out through this channel that's in the basolateral side. And then so I think this was talked about earlier too, but basically like this like pathway with Wink and SPHAKOSR1, it's like very important in the distal tubule. The distal tubule can be like regulated by like hormonal pathways. So we have like the kinases WNK, they can phosphorylate SPAK and OSR1, that activates them, and then they go and activate NCC, and that would increase salt reabsorption. But let's say you want to decrease salt reabsorption, there's like a few ways to do that. let's say you want to have like a really concentrated urine, you don't want to reabsorb that much salt, you could use this KLHL3. This is a ubiquitin ligase, so it inhibits the kinases here. And if these are inhibited, then the rest of the pathway is not going to work. So now that this entire pathway is inhibited, this is NCC is not going to be activated anymore, so you're not going to have that much salt reabsorption occurring. And then the next thing is, next way that you could kind of like kind of inhibit NCC is through thiazides. So thiazides are this diuretic drug that you use to treat blood pressure and inhibit like salt reabsorption. So basically thiazide just like targets NCC. If you have less water reabsorbed, then you're gonna have less salt. If you have less salt reabsorbed, then you're gonna have less water reabsorbed, because water likes to follow salt. So if you have like less water reabsorbed, that means there's more water in the urine, and that means you just increase the volume of the urine, and that means that would cause like your blood volume to decrease as well. So if you use thiazide, you can treat like high blood pressure, because it's going to increase the volume of urine, which should decrease the blood volume. And then the last thing we'll talk about in this topic is the RAAS, the renin angiotensin aldosterone system. So you guys really, you should kind of like, maybe if you can memorize this picture, because it's like really important to know this entire pathway. So basically the system is in effect when we have like low blood pressure. So if we have low blood pressure, there are some sensors in the kidneys that can sense this. So when they sense this drop in blood pressure, you're going to have the kidney release renin. It's going to increase like the release of renin, and renin will cleave angiotensinogen into angiotensin 1. Angiotensinogen, it comes from the liver, but it's mostly like circulating in the blood plasma. So renin will cleave it into angiotensin 1, and then an enzyme called ACE, it comes from the lungs. It will convert, as you can see, like it's called angiotensin converting enzyme. So the name kind of tells you what it does. It just converts angiotensin 1 into angiotensin 2. Angiotensin 2 has a lot of different targets. And all these targets are like, they work in tandem kind of to increase blood pressure, because this system only like is in effect, it only starts to, it's like, it starts to work when you have low blood pressure. So you're trying to increase blood pressure. So angiotensin 2, it can target the brain and cause you to become more thirsty, because if you drink more water, you increase like the blood volume. It can also increase your vasopressin secretion, or ADH. And ADH, like, it works to cause, it like basically tells the kidneys to reabsorb more water. So once again, you would want to reabsorb more water because you want more water in like your blood plasma, so that you could increase the blood pressure. It can also constrict your blood vessels. And this is done to kind of like maintain whatever little blood pressure you have left. And then it would also target the kidney to retain more salt. Because if it reabsorbs more salt, then more water will follow and then like be reabsorbed as well. That would increase your blood volume, increases blood pressure. But a really big point is that angiotensin 2 also targets the adrenal cortex, which sits on top of the kidney, and it makes the cortex, it releases aldosterone. So aldosterone goes to the distal tubules and it regulates the entire wink slash like OSR NCC pathway, the pathway that we just talked about in the distal tubules. So basically it up regulates that pathway, which means like NCC is going to be activated, and that would increase salt reabsorption. And like I said before, if you increase salt reabsorption, water is going to follow, so you're going to increase water reabsorption, and then your blood pressure can finally go back up. So one more thing is that like there is if you do have this entire pathway is just to deal with low blood pressure if your blood pressure drops. But if you do have high blood pressure, to prevent this like pathway from occurring, a lot of like drugs are ACE inhibitors, and they will target ACE, inhibit it, so then like angiotensin 1 can never become angiotensin 2. And angiotensin 2 is like the main like hormone that creates all of like the effects that are going to raise blood pressure. I think that's it for me.
Yeah, I think the next is my portion, so I'm gonna try to share my screen. Are you guys able to see this now?
Yeah.
I'm gonna take that as a yes. Okay, so topic 9 focuses on the loops of Henle, how the loops of Henle regulate final osmolarity of urine, how ADH plays a role in concentrating urine, and how ADH controls aquaporins in the collecting duct. And a variety of animals have proximal and distal tubules, but only mammals and birds have the loops of Henle. And here is just kind of a diagram of, I guess, the nephron. So it starts with the Bowman's capsule and it goes into the proximal tubule, the descending limb, then the actual quote unquote like hairpin loop, the ascending limb, the distal tubule, and then the collecting duct. And the purpose of the loops of Henle is to make highly concentrated urine, which helps animals conserve water or reabsorb salts. And we have about 1.2 million of these nephrons per kidney, and many nephrons can empty into the same collecting duct. So one point that, can you guys see like this side where I'm circling around? Half my like screen is blocked by Zoom, but like I don't know if you guys can still see the words of the slide.
We can see the whole slide. I don't know what
Yeah.
you're circling though.
Okay, perfect. So the loops of Henle, this is like something that Dr. Cho emphasized. They're most shallow in a beaver and they go deepest in desert gerbils, which means that species that live in dry environments have the longest loops of Henle. And that's because a longer loop gives a greater ability to make a more concentrated urine. You cannot actively transport water, which is an important fact to remember, that water will always move passively. So your kidneys do filter ECF and they do reabsorb water, but they do this by generating an osmotic gradient that allows water to flow out of the collecting duct. So basically, like as the collecting duct goes like deeper and deeper, there's a higher and higher osmolarity that allows water to be reabsorbed. And ADH helps regulate how much water can be reabsorbed by regulating aquaporins. And if you want to make a more concentrated urine, add more aquaporins to the collecting duct to reabsorb more water from the collecting duct. And the max human osmolarity is about 1200 milliosmolar. And the loops are what actually make the high osmolarity down here that allows water to be reabsorbed from the collecting duct. And then so a closer look to inside of like what one of these loops actually looks like. This is kind of just like a zoomed in version of like, I guess this portion or somewhere around here. So the descending limb, it's highly permeable to the passive transfusion of sodium chloride, which will flow into the descending limb, and water will passively flow out of the descending limb. And then to circle back up here, the ascending limb has low permeability to water, so not much water will be able to flow in or out. And it has active transport of sodium chloride out. So that is meant to increase the osmolarity in this area. And another point that Dr. Cho mentions is the ascending limb has a lot of active transport. So he like calls it the thick ascending limb because it has also a lot of mitochondria to do all this active transport. NKCC2 in the ascending limb is a secondary active transport protein, which uses the sodium gradient to pull in 2 chloride and 1 potassium into the ascending limb cells. And the difference between this and the ascending limb can be up to 200 milliosmolar. So if you go down this, it'll never be greater than 200 milliosmolar between like the descending and the ascending loop or the ascending limb. Osmolarity increases as you go deeper into the medulla. So up here in the cortex, it's not very, like the osmolarity is not very high. And then as you go deeper and deeper, the osmolarity increases. And there is constant active transport in the ascending limb, which helps magnify the gradient. This gradient is always present. It is never the same like osmolarity all throughout. And even when you don't need to concentrate the urine, there's still this gradient present. And water transport is regulated in the collecting duct, which is this area, which helps achieve the concentrated or the dilute urine, which is also regulated by ADH through aquaporins. So diuresis and antidiuresis are both regulated by ADH. Diuresis is a high urine output with a low urine osmolarity. So this would probably be something where you drank like a lot of water and there's low ADH secreted during this time because you don't want to reabsorb so much water. And then antidiuresis is a low urine output and high urine osmolarity. So this would probably be something where you like ate a ton of salty food that day. And a lot of ADH would be secreted during this time because you want to reabsorb that water from your collecting duct. And the posterior pituitary is what releases the ADH, and it can also be known as AVP. And ADH works by binding to a GPCR in the basolateral membrane, which you should also recall from the first exam how a GPCR works and how that cascade kind of progresses. But this cascade ends up phosphorylating aquaporin 2 to insert aquaporins into the membrane. And I believe they get inserted into the apical membrane. And this all occur like this whole process is translocation because the aquaporins are stored in vesicles. And then they're like translocated to reabsorb more water. And If ECF osmolarity increases in the blood, this increases ADH secretion, because if we increase ADH secretion, we reabsorb more water. Things like breathing, because when you breathe, you actually breathe off like some evaporation of water. Sweat, ingesting something super salty, will increase your ECF osmolarity. And salt reabsorption promotes water reabsorption, which makes less urine and raises blood pressure. And this is also just more diagrams of like how like this would be anti-diuresis because it's a very high osmolarity urine being produced, and this would be diuresis because as we can see, like the osmolarity is really low compared to anti-diuresis. And then Dr. Cho talks about 2 different medications, but both of these medications lead to diuresis. It just leads to diuresis through different pathways. Furosemide leads to diuresis by inhibiting NKCC2 in the ascending limb, and thiazide causes diuresis by inhibiting NCC in the distal tubule. And he talks about urea at the end of topic 9, which is a nitrogenous waste product made in the liver from amino acids. It can be used to make more concentrated urine, which is really like the most important point, is that urea can make more concentrated, like higher osmolarity urine. Urea can be partially reabsorbed, especially during antidiuresis, and urea is most permeable in the descending limb and the collecting duct. And that's it for my review of topic 9. I believe it's questions next. Yeah. So.
All right, sure. Okay,
I'll stop.
Okay, so oops, you guys didn't see that. Okay, so number 1 and number 2, go ahead and give it a shot. So I guess I'll wait like maybe around another minute or so. Sure, yeah, what do you think you got for number 1? Type it in chat if you guys want. All right, so Nikolai said he thinks number 1 is B, and yeah, that is true. Let's talk about why. Once again, the only one that's really dependent on, another transporter is this SGLT1, because once again, this is an example of secondary transport. So obviously it depends on this gradient being here. And what creates that gradient? Remember it was this sodium potassium pump. We talked about it generates your membrane potential, and it also generates the concentration difference between sodium and also between potassium. And then, yeah, you got it right for number 2 as well. It is C. Even though I said that facilitated diffusion is saturable, you can shift this curve upwards. All you would have to do is import more transporters into the membrane. So that is correct. Let's look at A. Increasing the concentration difference would not increase the net flux because remember, all the bus seats we're taking, all the enzyme active sites are already saturated. B, this is not possible. Well, like we just talked about, you can shift this curve simply by importing more transporters. And then D is also wrong. So good job. And then Number 3 here. Which of the following statements properly describe the graph above? And like last time, you can just drop it in chat if you want. All right, Nikolai said E, and I agree. So why? Well, let's look at the graph. First thing we can see is that it looks like whatever's being transported is not able to transport in the first half of the graph. What can we cross out? We can cross out simple diffusion. You cannot control what diffuses across a membrane. It will just happen. Or in simple diffusion, things will just move across the membrane. So that's why we can eliminate choice B. And then what else do we notice? We notice here that the transporter, whatever is being moved across the membrane, eventually reaches equilibrium. So that will eliminate active transport, and it will leave behind to us anything that is passive and that is not simple diffusion. So there you go. We have voltage gated ion channel. These are able to be regulated. There's this table in the notes. You should be able to fill this out. As we can see, simple diffusion, you can't regulate that. However, you can regulate channel diffusion and you can regulate facilitated diffusion. They are both proteins that you can modify. And then circling it back around, if you remember, in that pyramid of modifications that can occur, this type of regulation would be considered an acute change, something like phosphorylation or like ligand binding, things like that. And then I have 2 more questions here. Anyone have any guesses? All right, Nikolai says 4 is E. Yep, and I agree with that. Simply by looking at the concentration and then the charge of the cell, you can cross out A and B for choices C and D. Let's think about it. Let's look at A minus here. At equilibrium, or let's look at the forces directing it. Obviously, the charge is repelling it. So electrically, it's being pushed out. Chemically, it's being pushed in. And so the reason why A minus will actually have a higher extracellular is because it's unable to basically A minus, if the concentrations were equal on both sides, there will still be a negative charge on the inside of the cell. Because of this, there will always be that electrical force repelling it. So even if A minus managed to reach equilibrium chemically, there will be a charge pushing it outside, which is why it will have a higher extracellular concentration at equilibrium. And we can use the same logic to determine that for X plus, that it was going to be higher on the inside of the cell, actually. Okay, and then what about 5, Nikolai? What do you think? See? Yes, perfect. All you had to do here was we added up the concentrations, converted it to milliosmolarities. And just be sure that if I actually put NaCl here, you would have to multiply 80 by 2, multiply 200 by 2 as well before adding it up. Okay, so yeah, those are all the questions I had.
Should I screen share or do you want me
Whatever you want to, I can just keep it here.
Yeah, that's better. Yeah. Right. So this is one of the topic 7 practice questions. So I'll give you guys around a minute and a half to answer this one. It's kind of hefty, so yeah. Anyone have any guesses so far? If not, I'll give you like more time, so like 30 more seconds, maybe. Right. Ava said D, and that is indeed correct. Where is Ka? Can you go to the next slide by any chance? All right, thank you. So yeah, so the question is, the cell circle is filled with solute A, and the bath is filled with solute B, a different molecule, of the indicated total osmolarities, right? So after the cell is placed in the bath, it shrinks with time. What best explains this shrinkage despite equal starting osmolarities? So the big key thing that you have to know is that the cell is shrinking with time, right? So if it's shrinking with time, what is happening? Water is moving out of the cell, and therefore solute A is essentially moving out of the cell into the solution. So, but in order for the cell to shrink, the movement of solute A has to be greater than the movement of solute B from the solution into the cell. So that means it's going to be a net movement of solute A out of the cell because it's greater amounts of solute A moving and greater amounts of solute B moving into the cell. So if that were to happen, permeability of solute A out of the cell has to be greater than permeability of B that moves into the cell. That's why this question is D. Why the other choices are wrong? So first of all, water cannot be actively transported, right? It can only pass through the membrane through osmosis, so simple diffusion. And in the case of B, so that's the complete opposite of the correct answer, so that's why that one's incorrect. And solute A is actively transported out of the cell. In this case, solute A is not being actively transported. It's actually being passively transported because it starts from an area of high concentration within the cell and it's moving out of the cell into the solution, which has no sol UA. So essentially, because it's going from high to low concentration, it's going to be passing through either facilitated diffusion or through simple diffusion. All right, I think you go on. All right, this will be the next question, another hefty one. So I'll give you guys around a minute for this one. Anyone have any guesses? Remember that a vertebrate mammalian cell has an osmolarity of 300, so that can help. Anyone? No? All right, if not, we can just go on to the correct answer, which in this case is C. So the question is, a vertebrate mammalian cell is transferred to a concentrated solution containing 100 millimolar glucose and 200 millimolar NaCl. What acute regulatory responses does it activate? Well, so the first thing you have to determine is essentially whether the cell is shrinking or essentially where the water movement is going, right? So in this case, we have to determine the osmolarity of the solution. So we're given that it's 100 millimolar glucose and 200 millimolar NaCl. So remember the osmolarity of this NaCl would be multiplied by 2 because there's 2 moles of solutes. So in this case, it would be 400 milliosmol plus 100 milliosmol. So in that case, it's going to be 500 milliosmolarity for the solution. And the vertebrate mammalian cell has a milliosmolarity of 300. Because the milliosmolarity of the solution is higher, that's going to cause water to move out of the cell into the solution. And what occurs? Cell shrinkage. And this question is actually asking what is the compensatory response that occurs in response to the cell shrinking. So for this case, you want the cell to go back to its normal shape. So you would want water to move into the cell. And so for water to move into the cell, solutes have to enter into the cell. And the only pathway that does this is through the phosphorylation of NKCC, right? By phosphorylating NKCC, you activate this co-transporter that essentially allows solutes, sodium, potassium, and chloride to enter the cell. And if those solutes enter the cell, water will follow. And because of that, the cell will swell up and return back to its normal shape and essentially counteract that initial shrinkage. The other choices are wrong because, first of all, PASC OSR1 does not dephosphorylate, it phosphorylates. So anything that says dephosphorylates, you can eliminate. And in terms of these answer choices, phosphorylation of NKCC does not cause solutes to leave the cell. It actually causes solutes to enter the cell. That's why this one's wrong. And for this one, PASC OSR1, so the phosphorylation of KCC does not cause solutes to enter the cell. It actually just inhibits KCC from allowing solutes to leave the cell. So that's why that one is wrong. Yeah. Last question. I'll give you guys around or a little less than a minute because this one's a little less hefty. So. Yes, the answer choice is indeed D. For this case, you have to remember this formula on the bottom right there where it says ingestion plus metabolism equals excretion, egestion, and evaporation. Right. So the big thing about terrestrial animals is that they don't have the capability to do absorption like aquatic animals because they lack gills, right? Because they lack gills, they are not in direct contact with their aquatic environment. Therefore, they cannot exchange contents and fluids with through their gills, right? So anything that says absorption, which is 3, you can eliminate those choices. And in this case, ingestion, metabolic water, as I mentioned, is equal to excretion, egestion, evaporation, which is choice D. So, yeah.
I think Nikolai had a question.
Yeah.
Did you have one?
Sorry,
The mic is really distorted. We weren't able to hear you. Or understand you.
Is that better? You like it more? I'll type it.
Yeah. Could you type it?
Sorry.
Yeah, sorry.
Are you sure? All right.
Okay, now we'll go through my questions. Okay, we'll start with like the hemorrhage questions. So basically, I'll just read them out loud. So Sally suddenly experiences severe blood loss. Basically, something happened, she got injured in some way, and now she's hemorrhaging. So how is her initial ECF osmolarity going to be affected by this? And you guys can just type it in the chat. Yeah, it's C. So you can click. It should. Yeah. Wait. You can go back. Okay. So, so basically, her initial ECF osmolarity will remain unchanged, and it's because like when she's experiencing like severe blood loss, it's just that she's just losing volume. There's no like solutes or anything being added to it, so it's just her initial ECF osmolarity that's going to be affected by this. For the next question, how will Sally's blood pressure be affected from the hemorrhage? Since we kind of already showed the answer, I think I'll just like go over it really quickly. So, the answer will be that her blood pressure will drop. So, okay, so her blood pressure is going to drop. Like I said in for the first question, she's going to lose a lot of blood. So her blood volume is decreasing. So blood volume and blood pressure, they're kind of like related to each other. Well, for this like exam, this class. So basically, if your blood volume is dropping, then your blood pressure is also dropping. Next question. So Sally visited Daytona Beach and decided to swim for a little bit. She's not the best swimmer and she keeps ingesting the seawater. So what happens to her initial ECF osmolarity? So as you guys saw, it increases. So when she's ingesting this, when she's ingesting the seawater, she's taking in a lot of solutes. She's taking in a lot of solutes from seawater. So her her concentration of her extracellular fluid is also increasing because you're adding solutes to it. So the last question, how will Sally's blood pressure be affected from her ingesting the seawater? You guys can answer this question by typing it in the chat. Okay. Okay, yeah. Ava, yeah, she's right. It is A. Her blood pressure will increase because when she's ingesting the seawater, like I said before, you're increasing the solid concentration of the extracellular fluid. So if that's happening, then that means there's a lot of solute in that fluid, but there's also the fluid that's inside the cells. That's the intracellular fluid. That fluid is going to move out of the cell into the ECF osmolarity to like balance the high concentration. To balance like the high concentration. So that means more blood is going into the ECF. It's flowing out of the cells into the ECF. So the volume is increasing. The volume of water. So if the volume of water increases, her blood pressure also increases. Okay, that's it for my questions.
Okay, so my questions are pretty like short and straightforward, so I'll just give you like 30 seconds to think about number one and drop an answer in the chat if you want to participate. If not, we can just like wait. Right, well, Nikolai seems to be on it. Whoever's controlling the slideshow, do you want to like press? It is B. If you are super sweaty, for number one, it's B. Because if you are sweating, when you sweat, you like mostly excrete water more so than solutes. So that would cause your ECF or like your osmolarity to increase because you're excreting a lot of water and not a lot of solutes. Um, so if your ECF increases, then you want to reabsorb more water from your urine because you want to like lower that ECF a little bit. And if you increase ADH, then that increases aquaporins, which would then increase water reabsorption to help kind of offset the fact that like you're releasing all this water through sweat and not releasing as much solutes. And then for number two, oh, well, I guess the cat's out of the bag for number two. It's most active during antidiuresis because urea can be used to increase the osmolarity of urine. Okay, so I'll give you guys 30 seconds for number three. So yeah, three is pretty much just recall. Furosemide is inhibits NKCC2 in the ascending limb. But I do remember a few questions from my exam on like the thiazide and furosemide and how they impact the body. So I figured I would do a question on that. And then if you guys want to think about four and drop it in the chat as well. So yeah, Ava, you're correct because for number four, the answer is A. it would have no impact on ADH release because for a blood transfusion, you're gaining blood volume, but it's the same like osmolarity. So your body wouldn't try to like make your urine like more or less concentrated because you're in like the fluid that's being put into your body is the same concentration. So your osmolarity kind of stays the same. And that's the end for my practice questions.
And now I guess we have like 10 minutes for some questions. Oh, Keandre, I think Nikolai typed his question out. You saw that.
Yeah, 1 second, let me read it. First question was because eventually active transport would surpass the equilibrium. Over a longer period of time. Okay, so essentially active transport, as I mentioned, it's not going to be actively transporting A down outside of the cell because essentially it's moving down from a high to low concentration. So it's going down its concentration gradient. Remember, active transport is essentially
But it's still like it's still happening. It's just continue, right? You have like this fall back in the same that you have active transport. It's just after it can continue after asking.
okay. Well, right. So for this case, which it's asking what best explains why so essentially you're not wrong. Indeed, you're not wrong. But in terms of this question, the best answer would be that the permeability of solute A would be more than solute B. So I guess I should have considered that in terms of it could surpass equilibrium. In that case, it would actively transport it back. But for this case, the best answer would be permeability of solute A is more than solute B. So
Yeah, I didn't mean it as like a gotcha. I just wanted to make sure I had it right.
Right. So typically he had like a clicker question on this, and it's he asked it in the form of like swelling with time. So the correct answer was permeability of solute B is more than solute A. So this is essentially the complete opposite because you're talking about cell shrinkage, and for cell shrinkage to occur, solute A has to move out, and therefore water will follow, and therefore the cell will shrink. So in this case, it's the complete opposite. But yeah.
Like,
Yep. Is there any other questions? If not,
I guess if not, we can just.
can't really type this that fast, so sorry.
Oh, okay. You have another one? Okay, sure.
Well, like means the builder like not simply their flow rate and parking just accidents like distance.
Sorry, it was really hard to understand you again. I think you said
Honestly,
something about flow rate.
it's I'm still like a bad rule and break it.
Are you talking about like the GFR stuff slide?
I'm gonna type it.
Okay. Oh, okay.
Is this wait, is this a topic 8 or a topic 9 question? Okay. So here, can whoever's controlling one more slide. So each part of the kidney tubules. Basically, like the main thing that's changing in like your urine concentration isn't so much of like what's in each part of the kidney tubules. It's mostly happening in the collecting duct. The collecting duct is like what really has the most dramatic changes during like diuresis and antidiuresis, and that is just because of like water reabsorption due to ADH. Like there might be like less dramatic differences throughout like the descending limb and the loop and the ascending limb as far as like the osmolarity differences. But the biggest thing that's going to change is the flow rate in the collecting duct of water. Like, so if you're in diuresis and you have a really high urine output, you're not going to reabsorb as much water. I don't know if I'm addressing your question well enough. Like, please feel free to like comment at any point. But for antidiuresis, um, you're going to reabsorb most of that water in the collecting duct, so your flow rate, your urine flow rate will not be as high, but like your reabsorption rate of water will be high for antidiuresis in the collecting duct. I don't know if that helped, Nicola. Okay. If not, feel free to come to office hour. You're welcome. Any other questions?
All right. If if not, that's all we had. Thank you for coming. Good luck studying. We'll post the slides and the recording as well.
Danae, you there?
Isn't he?
Idney, Danae, Idney, which one? um, I guess you can just end the recording now.
Okay.